A very good morning, sir. My name is Imalina Liu. Today, I will be presenting on fund implementation, which is a case study. Now, I will be presenting on the introduction of this study case. First of all, the harvest. This is the first step of manufacture of wine. The second part of manufacture of wine is crushing and pressing. The third step is fermentation. Fourth step is clarifications. Once the process of fermentation is settled, the clarification process starts when winemakers have decided to rack or to siphon their wines from one tank or using a barrel. The process of winemaking is involved in aging and bottling. After continue on, I will be continue with our content, my content study case questions part 1. The first question is, what chemical process is needed to make wine? Fermentation is a chemical process by which molecules such as glucose are broken down anaerobically. The second question is, what is an inoculum? Inoculum is a small amount of bacteria or microorganism that used to start a cell culture. It is a material used for inoculation in winemaking. This is one of the most important elements to make great wine. These will convert to sugar of wine grapes, becoming alcohol and carbon dioxide through the process of fermentation. When there is absence of oxygen. So fresh yeast is better at metabolizing glucose to yield the amount of correct ethanol. That is the reason why. The yeast that Stanley used was living. This is because there was evidence that showed the yeast was able to metabolize. The next morning, Stanley saw that the bubbles covering from the top of the mixture in the glass, the bubbles showed that it produced carbon dioxide. Thus, some glucose had already been metabolized. Part 2, which is for the study case question. I will be briefly explaining that the difference between yeast and bacteria. So, yeast is a microscopic fungus consisting of single oval cell that reproduce by budding. Meanwhile, bacteria is a member of a large group of unicellular microorganisms containing a cell but is lacking of organelles and an organized nucleus. So, the main reproduction method is asexual reproduction, which is binary fission. So, yeast, budding, and bacteria binary fission. Meanwhile, yeast converts sugar into alcohol and carbon dioxide, which is, we all know, known as fermentation. Thus, in production of beverages, yeast has a more important role. Yeast has an important role because of its ability to accumulate high levels of ethanol. Moreover, yeast can produce high aroma compounds. Meanwhile, for lactic acid that's from the bacteria is also important in the process of fermentation because they produce peptides, which will inhibit growth of unwanted organisms and also create flavor compounds. So pure yeast will not make the wine sour. This is because it produces, as I said from the slide before, it produces high aroma compounds but not flavor compounds. Bacteria can convert pyruvate to acetic acid that will not make the wine have sour taste. Thus, adding yeast does not make alcohol become sour. So the difference between an obligate aerobe and obligate anaerobe and a facultative anaerobe depends on an aerobic respiration because it uses a terminal electron acceptor, while an obligate anaerobe depends on anaerobic respiration. You see, it's connecting. So using a final electron acceptor other than oxygen and also depends on the process of fermentation. Meanwhile, facultative anaerobes will show better increase in growth in the presence of oxygen, even though it will also grow without it. Therefore, yeast is an organism known as a facultative anaerobe because it can survive both conditions, which are aerobic and anaerobic. Stanley didn't make wine. He already exposed the yeast using an open container which exposed yeast to the air, and it of course did not ferment. Furthermore, bacteria would already contaminate it and yeast was not fresh. A pure yeast culture is important to avoid contamination with bacteria or other microorganisms. The third part of the study case questions. This is the summary equation for cell respiration, as you can see. So there are 3 stages, which is glycolysis. The second one is Krebs cycle or citric acid cycle. The third one is oxidation phosphorylation. Aerobic respiration, I've made a table so it will be easy to see the difference. So NAD plus is very insignificant in fermentation because it allows glycolysis to continue to produce ATP. Without NAD plus, cells can go through glycolysis and the production of ATP stops. Furthermore, the significance of NAD in cell respiration is a coenzyme, which is an electron carrier or acceptor during cellular respiration. When oxygen is not present or if an organism is not able to undergo aerobic respiration, pyruvate will undergo a process called fermentation. Fermentation does not require oxygen, therefore anaerobic. Fermentation will replenish NAD from NADH and hydrogen produced in glycolysis. Fermentation will produce more smaller amount of ATP that will ensure survival. Stanley needs to use a closed container, in my opinion, to prevent any presence of oxygen because to use pure yeast to avoid any contamination, and lastly, to use juice that has been sanitized to eliminate bacteria. With part 4, in hypothesis for Stanley's experiment. All juices will ferment at the same rate if the juices have the same sugar content. This is what material Stanley needs to carry out for his experiment. In my opinion, he needs yeast, test tubes with corks to prevent presence of air, water, full juices, and a Brix meter. This is the reason why. This is an experimental protocol that includes a control experiment. The last question in part 4 is identify the following components of your experiment. A dependent variable is the different types of juices. Dependent variable is the rate of fermentation. Control variable is the volume of yeast. Part 5. This is the chart that I have made to compare my experimental design from Stanley's design. So for the hypothesis, materials, procedures, and variables. And based on both experiments, the similarities between both experiments are the hypothesis, which is all juices will ferment at the same rate if they have the same sugar concentration. Other than that, the similarities between both experiments is different types of produced for the independent variable. The dependence variable in both experiments are the same, which is to identify the rate of fermentation. The control variable is also same in both experiments as they use the same amount of yeast for each solution. The differences between both experiments is the materials to conduct the experiment. Based on experiment part 4, only yeast, test tubes with corks, fruit juice, and Brix meter are provided, while in experiment 5, there are more than 4 materials needed for the experiment. Then the procedures in experiment 5 are more detailed and specific than the procedure in experiment 4. Based on Stanley's experiment that I have learned, the amount of juice added for each fermentation tube was different. The reason behind this is because the amount of sugar will vary in each juice, so different amounts of juice in each fermentation tube in order to have a total of 2 grams of sugar. The positive control for Stanley's experiment is when the weight of sucrose is the same. Negative control is the experiment is that the different volumes of water. I would measure the displacement of liquid to determine the volume of carbon dioxide produced. Part 6. These are the questions for part 6. This is the last part of the study case questions. The results shown proves that the control experiment will help to increase the accuracy of results. Second, based on the result, the volume of carbon dioxide produced for each juice is the same when the sugar concentration is similar and same. Thus, the hypothesis is accepted. Firstly, the displacement of liquid. Second, the volume of carbon dioxide produced. Thirdly, the sugar content. And fourth, concentration of yeast. Based on the results, strawberries are preferred by yeast of fermentation. This is because the sugar content of strawberries is similar to the sugar content of grapes. In my conclusion, there are factors that will affect the rate of fermentation, such as sugar concentration and the displacement of liquid. The process required for fermentation is very detailed and must be accurate. Therefore, Stanley's experiment will succeed and will help you understand better on the process of winemaking. These are the references that I've provided. name is Emmanuel Liu binti Ismail Liu, and that's all from me. Thank you.